Welcome back from the break. We're on the home stretch now. Our last session of presentation starts with submitting an eCTD, most common submission issues, and FDA plans for eCTD version 4.0. Our first presenter, Jonathan Resnick, is a member of CDER's Division of Data Management Services and Solutions. He focuses on electronic submissions, and he's been with the FDA since 2011. Prior to joining FDA, Jonathan spent 18 years working in IT to support federal and private sector clients. Our next presentation is on the CDER NextGen portal. What's new? Our presenter for this topic, Mr. Seyoum Senay, leads FDA's mission-critical informatics initiatives in support of human drug regulatory review processes with a customer focus through analysis and pragmatic solutions that work to advance CDER's public health mission. Our third presentation is on the impact assessment of the 2021 Data Integrity Notifications to Sponsors. Our presenter, Dr. Nilufer Tampal, is the Associate Director for Scientific Quality and OB within OGD. In this role, Dr. Tampal develops strategies and oversees implementation of data quality and the scientific integrity of bioequivalence data submitted in ANDAs. She provides leadership and expertise in utilization of advanced analytic tools in the assessment of bioequivalence studies submitted in ANDAs. Next, we'll present on risk factors for benzene contamination. This presentation will be delivered by Dr. Pallavi Nithyanandan, who's the Director of the Compendial Operations and Standard Staff, Office of Policy for Pharmaceutical Quality, CDER. She's been with FDA since 2010, serves as one of CDER's experts on compendial issues, and is the CDER delegate to the United States Pharmacopeal Convention. Our final presentation is on approaches to mitigate the risk of nitrosamine impurities in pharmaceuticals. Our presenter, Dr. David Curie, is the Director of the OPQ Office of Testing and Research and has used his analytical skills to address issues that arise with complex drugs such as heparin, glatiramer acetate, transdermal systems, inhalers, modified release dosage forms, and protein-based therapeutics. Please join me to welcome our first presenter, Jonathan Resnick.
Hello, my name is Jonathan Resnick. I'm with FDA CDER in OBI in the Division of Data Management Services and Solutions. And today I'll be talking about submitting an eCTD, the most common submission issues in FDA plans for eCTD version 4.0. So in today's presentation, we'll be covering a bit about guidance, the most common reasons for technical rejection notices, eCTD validation as it pertains to submissions containing study data, frequently asked questions to our electronic submissions help desk, and FDA plans for implementations of the eCTD 4.0 standard. Let's take a quick look at eCTD guidance. We know that industry must follow FDA guidance when submitting an ANDA. The details are in the FDA 745A guidance called Providing Regulatory Submissions in Electronic Format, Certain Human Pharmaceutical Product Applications and Related Submissions Using eCTD Specifications. And we have a hyperlink right here to take you directly to that guidance. There are two main pieces of it. The first, submissions must be in eCTD format. And the second, submission size 10 gigabytes or less must be submitted through the FDA Electronic Submission Gateway. If these requirements are not followed, the submission will be rejected. I've included here 4 key pieces of information that you can use as resources related to requirements and guidance. So the first is that eCTD guidance currently on revision 7. The second is the eCTD Technical Conformance Guide. The third, the eCTD Submission Standards, and here I've provided a screen capture of a page on our FDA.gov website which contains the submission standards for eCTD version 3.2.2 and Module 1 version 3.3. And finally, there's a link for our eCTD website. So let's take a look at the most common reasons for technical rejection notices. Once the submission comes into the center, there are a few basic validation checks that happen prior to the main eCTD validation run. What we've noticed is by far and away the biggest failure in the validation check is for duplicate eCTD sequence number. We've noticed it accounts for nearly 50% of all eCTD rejections from the center. Passes these basic validation steps. We then use our eCTD validation tool. And what we find here are some of the top eCTD errors that result in an auto rejection. So let me go through these with you. The first one is that all the submitted files were not referenced in the backbone. And that's error code 1306. So what this is all about is if you provide a file in the eCTD sequence folder, but that file is not directly referenced in the index XML or the US regional backbone files, then it's not going to be displayed to the reviewer when they're using the eCTD viewer tool. That's why this is such a big problem and why a submission will be rejected if the file is not referenced properly. The second top reason for rejection is error code 1323. And in many ways, this is the reverse of error 1306. This is where the submitter has referenced a file in the backbone so that the reviewer, when they're using the eCTD viewer tool, they see a document listed on the screen. But when they click on the document, it says file not found, because the actual file itself wasn't included in the eCTD sequence folder. Then the third top error is unsupported DTT version used in a US regional XML. Not too surprising, since last year was when we stopped supporting that old Module 1 DTD 2.01 version. Then we have an error called ts.xpt with information on study start data for each study is not present. That's error code 1734. I'm actually going to get into quite a bit of detail on this particular one. This has to do with the technical rejection criteria for study data that FDA implemented back in September of 2021. And we're going to take a good look at that to help make sure everyone understands what's going on there. And then finally, in terms of auto-rejected errors, all files provided in the study section must be referenced by an STF or study tagging file, and that's error code 1789. I also have a couple of additional ones down here. These are top errors where manual review happens. So this one would be a submission type is invalid for an application type, or a submission subtype is invalid for submission type and or application type. One thing to keep in mind when looking at these rejection numbers and when we're talking about top errors is that for 2022, FDA rejected less than 2% of all eCTD submissions. And just for kind of frame of reference, just in CDER alone, we received about more than 300,000 eCTD submissions. So when you see these numbers, although you might say, oh, 337 submissions, that's a lot, compared to the overall submission intake of close to 300,000, it kind of puts it in perspective. We also have this hyperlink down here to our specifications for eCTD validation criteria. That's where you can find out a lot more detail on these different codes. Now let's take a look at eCTD validations specific to study data. So, a few things. This past March 16th, we went live with a couple of updates to the FDA validation profile, and it relates to 4 validation codes. The first one is 1734. This is the one we had mentioned in that previous slide. And what's different about this code as of March 16th is it's no longer validating if the study ID in the trial summary file matches the study ID in the study tagging file. We moved that validation check to a new code, which is listed here, called 1738. Another change we did was with 1735. It's doing the same validation as always done in the past. The only difference is we've allowed for a condition where if the define XML file is tagged as data listing data definition, that it would no longer trigger 1735. So we allow that to be another valid file tag. Finally, on 1737, this is the same validation that's been in place for years. No change to the logic. The only thing is the validation is now looking across all study data sections in modules 4 and 5, with the exception of these five sections here. Previously, it was only looking at the TRC sections. Bit more on guidance here. There is a separate study data guidance called Providing Regulatory Submissions in Electronic Format, Standardized Study Data, last updated June of 2021. And here it says sponsors must conform to the standards in FDA Data Standards Catalog, and that applies to the NDAs, BLAs, ANDAs, and INDs. And here, in this conference, we're talking about ANDAs. Now, FDA is using eCTD validation 1734, 35, and 36 to confirm that sponsors are conforming to the FDA Data Standards Catalog. And FDA began using these validations and rejecting if they were to fail back in September of 2021. And this subset of eCTD validations are commonly described in detail in the technical rejection criteria for study data, also referred to as the TRC. And just a little note here, the technical rejection criteria for study data, you can find this within the Study Data Technical Conformance Guide. And you can find the Study Data Technical Conformance Guide along with the Data Standards Catalog under the Study Data Standards Resources page. And we've got a link here to make it really easy for you to find it. And then we have a couple of other links here, just helpful for you. If you needed to get some information in general about eCTD, here's the link to our eCTD website. And we also have a page for study data for submissions to CDER and CBER. Just some additional information about study data validations are located on here as well. On this slide, we took a look specifically at ANDAs during the years of 2021 through 2022. And we looked at ANDA submissions where study data was contained in those submissions and provided under the TRC applicable sections. And what we found is through the first half of 2021, there was quite a big failure rate. About 40% of ANDA submissions which contained study data in these TRC applicable sections were failing the technical rejection criteria. But as we got closer to September of 2021, which is when we had communicated that FDA would start rejecting submissions that failed this validation, we found that the conformance rate actually went way up. And so inversely, what you're seeing here is the failure rate of these validations went way down. So this red vertical line is when enforcement began, meaning when we started to reject if the TRC criteria failed. And you'll see that starting in 2022, the failure rates dropped dramatically. They're really just between 2 and maybe 5%, which conversely means that we're seeing a conformance rate, a successful conformance rate to the technical rejection criteria in the 95 to 98% range, which is really good. What we do here on this slide is we're taking a look now at that 2 to 5% of ANDA submissions last year that did not conform, that failed these TRC validations. And what we found is the TRC validations, number one, it's 3 validations, 1734, 1735, and 1736. And what we found here is it's 1734 overwhelmingly is the one that was giving people problems. It accounted for 58% of the errors, and the most common reason why 1734 was triggering was because the ts.xpt, or otherwise known as the trial summary file, was missing from the study. So we've summarized this information here in this slide. So to begin, let's just take a look again at what is the 1734 validation. It's a data set named ts.xpt with information on study start date must be present for each study in required sections. And the validation is looking, number one, is the trial summary data set present? If it is, study start date is provided. Study start date is in a valid format, and does the study ID in this trial summary data set match up with the study ID in the STF to which it's referenced under? And what we found is 54% of the cases where 1734 triggered is because there just was no ts.xpt file at all submitted for the study. The second biggest reason for 1734 to trigger is that if a TS file was submitted, it didn't have an accurate value here for the study start date, a valid value. And then finally, the third most common reason was that the study ID in the trial summary file didn't match up with the study ID that file was referenced to with a study tagging file. So now we're going to switch gears and we're going to look at a few frequently asked questions to our eSub help desk. We think these conferences are a really good forum to share some of the most common questions our help desk gets. So we've got 3 to go through. So the first one is, where should my document go within the eCTD structure? And what the team will do is respond back with 4 key reference documents. The first being the eCTD Technical Conformance Guide, which is a high-level guide on putting together an eCTD submission and how to organize it. And then the Comprehensive Table of Contents, Headings, and Hierarchy specification. This contains a list of all the predefined headings and subheadings in the eCTD. And then 2 ICH M4 guidelines, one called the Organization of the CTD, and the other one, the CTD Quality Question and Answers. The next question the help desk gets that we'd like to share is, why did my submission fail eCTD validation 1734? And the typical answer would be, well, study data was submitted to modules 4 or 5, and there's no valid trial summary reference under the STF for that study. And to see 8221 eCTD technical rejection criteria for study data within the study data technical conformance guide for details. So the study data technical conformance guide is the place to go to really understand 1734 and 1735 and 36, to understand how FDA is validating your submission that's contained study data with the eCTD validation criteria. And the last question we wanted to share with you is, will a validation error cause a rejection? And so here the help desk will respond back with a link to the specifications for eCTD validation criteria. So they can first provide that person with a list of all the validations. And then also point out that in that list, each validation will contain a severity description, and only the ones that are marked high are the ones that would result in a technical rejection notice. So now with the time remaining, I'm going to talk about FDA's plans for implementation of the next version of eCTD, that's version 4.0. So we begin by talking about the ICH level activities, right? Version 4.0 of eCTD was developed through ICH. ICH has published the version 4.0 implementation package. The latest version is 1.5 that was posted in May of last year. On the right-hand side is a screenshot of the ICH webpage for eCTD 4.0. So you can see that all of these documents are currently listed and available for download. There's also a Q&A change request document available as well. And you should also know that the different regions in ICH, so the different regulators, they have been posting information in a table on this webpage that will tell you when they plan to start accepting eCTD4 and when they plan to do technical pilots and so forth. As far as FDA-specific activities, FDA has published the eCTD4 Technical Conformance Guide and the FDA 4.0 Module 1 Implementation Package. The package was originally posted in February of 2020 with an FR notice asking for public comment. Since then, it's been updated a few times, and the latest version was posted in September of last year. FDA has also posted the specifications for the 4.0 validation criteria, comprehensive table of contents, headings, and hierarchy. And I've posted here on the right-hand side just a screenshot of a page that we've set up on our fda.gov website, which is dedicated to the eCTD 4.0 implementation, where you can find all of these documents. FDA is also continuously receiving software updates and testing to make sure that we're working closely with our own tool vendor to make sure that we can receive, validate, and view the eCTD4 submissions. And we're also in the later stages of a technical pilot to make sure that we're set up to meet the eCTD4 specifications. As far as our FDA implementation strategy, our plan is this. Our initial release is going to be to accept new applications in eCTD 4.0. It'll be voluntary. So the technical pilot that we're engaged in right now, that we're wrapping up this month, it's a small group and it's focused on just new applications submitted in 4.0. We're not talking about transitioning yet. We're also planning to provide the ability for industry to submit sample submissions in 4.0 for technical feedback. That would be available to everybody. We're planning to open the door to that later this year. We'll leverage our existing sample submission process that we have open today for 322 submissions. And then we're looking to accept these new applications in 4.0 either late this year or probably more likely early next year. Once we are accepting 4.0 submissions for new application types, we want to make sure everything is stable. So likely about a year after that, we'll look to begin accepting the transition of current applications. So applications that are in 3.2.2 today that you want to switch over to 4.0. And then once we have that implemented and stabilized, we'll then turn our attention to implementing two-way communication. A little bit of information here on the technical pilot. The objective is to determine if the implementation satisfies the requirements in the technical specifications and then make any changes prior to accepting 4.0 submissions in our production environment. And the testing started last year, last June, and it is wrapping up now. So how can you prepare for eCTD 4.0? We've put together 4 bullets here to really help give you a pathway. So first, discuss 4.0 development plans with your vendor or IT organization. Look to make sure you can understand the specifications. Is there a plan for transitioning? You can always send questions to ICH or FDA. Then become familiar with the eCTD 4.0 concepts and enhancements. ICH has put out a couple of PowerPoint slide decks to help you orient and understand the eCTD 4.0 implementation package. Take a look at our FDA 4.0 Technical Conformance Guide too. Then know where to find the eCTD 4.0 information, and I'm hoping this presentation here is going to go a long ways in helping you check that box. And then being able to submit a 4.0 sample submission for technical feedback. Later this year, make sure to check in on our eCTD sample submission process webpage because we plan to update that page maybe in the fall with instructions for how you can submit a sample 4.0 submission for technical feedback. And here I've included the particular link for the ICH 4.0 webpage and the link for the FDA's 4.0 webpage, so it's easy for you to find and bookmark. So in summary, we talked about common submission issues. Duplicate eCTD sequence numbers, they're the number one issue. We talked about the frequently asked questions to eSub. eSub at FDA HHS is a great resource. We talked about the ANDA study data conformance, how it's working out really well, that we're seeing 95 to 98% conformance in 2022 to these TRC validations. And when there is a problem with conformance, it's mostly related to validation 1734, and we looked at what are the top trigger reasons for that. And then finally, we talked about FDA plans for implementation of eCTD 4.0. The publications are out there. The specifications are published, rather. Testing is underway. And our sights are set to start accepting these 4.0 submissions either at the end of this year or more than likely early next year. That's the end of my presentation. Thank you very much for your time. I look forward to taking questions. Thank you.
Thank you for introduction. Hello, my name is Seyoum Senay. I am leading the FDA CDER Informatics initiatives in support of human drug application review and approval process with a customer-focused analysis and pragmatic solution that work to advance CDER's public health mission. Today, I will have the opportunity to speak with our stakeholders about the CDER NextGen portal. As many of you may be active participants in CDER NextGen portal, I will share how the portal have been leveraged to improve collaboration and operational efficiency the last year or so. So, agenda today, we will have overview of CDER NextGen portal. What's CDER NextGen portal? Before and after CDER NextGen portal, our digital transformation, and then what's new really during the last year or so. And we'll share our users' adoption success stories. So, what's CDER NextGen portal? CDER NextGen portal is a one-stop shop for the purpose of non-eCTD submission, collaboration, and reporting. With that, the FDA digital transformation is continue and provide value to receive clean, complete, and validated data to support CDER's mission. As you see, in the past, CDER used to receive paper and media submission through the document room. After CDER NextGen portal established, stakeholders come through the CDER NextGen portal, provide their submission, and that submission directly routed to the review division or internal workflow, and the reviewers can start their work as soon as the submission happened. We have CDER NextGen, we have about 20 portal products. Today, we'll go over the recent updates regarding CARES volume, control correspondence, and pre-ANDA meeting requests. So what's new? CDER NextGen portal recently released new product enhancements to support collaboration. The CARES Act being one, control correspondence and pre-ANDA meeting another. And then we also launched a pre-launch activities importation request. We call it Player. It's a new product supporting the Office of Compliance. CARES, we launched in March. The following updates were the CARES Act amount information reporting, processing. The updates include we have a new process to submit CSV files, separated files, which will address the previous limitation on the number of records in CVS file, and also addressed common file validation errors. We also added a new process to replace existing submissions and new functionality to view previously submitted reports. Moving on, for Generic Drug User Fee Act 3, we have a couple of enhancements in the control correspondence area, as you see in the screen. We have additional status type for pending for ANDAs. We also added screening questions to product details to align to GDUFA III commitments. We also updated certification text in alignment with Generic Drug User Fee Act 3 enhancement. In addition, on the right-hand side, we have a pre-ANDA meeting request as part of GDUFA III enhancement. We added to the meeting request addition of meeting types, product-specified guidance in alignment to the GDUFA III guidance. We also added additional questions added to the meeting request to align to the guidance. Next step, moving on to the pre-launch activities importation request or Player, launched in a February time frame. Now with Player portal product, stakeholders be able to submit a Player in preparation for market launch within 60 days of the anticipated decision date for pending initial original marketing applications. And this Player application or portal application is also enabled with two-way communication capability, so stakeholders and the FDA can correspond digitally, such as information requests, clarifications, final response letters. All this can be communicated in a two-way communication capability. So for this, the Division of Global Drug Distribution and Policy Import-Export Compliance Branch manages the Player requests and provide complete response to stakeholders' requests with review decision input from Office of Generic Drug, Office of Pharmaceutical Quality, and Office of New Drugs. Moving on, we have continued to see a growth in our user base. The CDER NextGen portal team can continue to make enhancements to improve user experience. As you see, we have 12,000 plus users on a daily basis accessing the CDER NextGen portal. So far, we have 100,000 plus submissions came through this pipeline. In summary, we have covered so far what's CDER NextGen portal, what's new, and user experience and adoption. So with this, we are continuing to provide you more updates, what's happening in CDER NextGen portal. And if you are looking forward to access the CDER NextGen portal, please go to cdernextgenportal.fda.gov and get your access and let us know your feedback. With that, that concludes my presentation for today, and thank you. You have a wonderful day.
Hello, everyone. I'm Nilufer Tampal, and I'm the Associate Director in the Office of Bioequivalence, Office of Generic Drugs. Today, I will be presenting our assessment of the impact on the stakeholders following 2 data integrity notifications from the FDA in September 2021. And what I'd like for you to take away from this presentation is the understanding that both sponsors and personnel at the testing site have a critical role in ensuring the integrity of the data. And both entities, therefore, need to consider appropriate risk mitigation strategies to address falsification of data in bioequivalence studies. Here's the outline of my presentation. I'll start with the background of the events leading up to the notification actions, and then we'll see how these actions impacted the patients and sponsors, and what were some of the lessons learned along the way. I'll also talk about the steps that can be taken to avert future problems of similar nature. So let's go over the background. In November 2019, FDA conducted inspections at the Panaxel and Synchron sites. One of the inspections was a surveillance inspection, and the other was a for-cause because the assessors had raised flags about the integrity of the study data in at least 2 applications. Following the inspections, the Office of Bioequivalence undertook extensive investigative analysis. And this was a very different kind of analysis, not something that we do on a routine basis. And with this analysis, we identified anomalous and unreliable study data from these 2 CRO facilities. We found instances of significant misconduct and violations of the federal regulations spanning multiple studies over a number of years across several drug products and involving multiple applicants. Consequently, general letters had to be sent to the CROs asking them to explain the observed anomalies, including the anomalies in the PK trend, as well as multiple instances of subject pairs with overlapping concentration profiles that we had seen during our investigations. And neither of the CROs were able to provide valid scientific reasons to rule out that the data were falsified. So then in September 2021, FDA posted the notification of actions to inform the stakeholders about the findings at these 2 CROs and also the action that the FDA was taking. The sponsors were informed to repeat the studies from the CRO facilities at alternate sites, and this included approved, tentatively approved, and pending applications for both the generic drugs and the new drugs. Also, since potential bioequivalence problems were identified, the approved products on the market were not considered to be therapeutically equivalent any longer, and meaning they were not interchangeable with the brand product. Therefore, FDA went ahead and changed the therapeutic equivalence rating of the affected generic products from AB to BX. Now, this notice also advised patients to continue with their treatment or at least not make changes to their treatment without consulting their healthcare professionals. And then the CROs, they were notified separately to rectify their system-wide failures with appropriate CAPA, the corrective and preventive action plans, so that the agency is assured of sustained compliance with the regulations from these two CROs going forward. This was not the first time that the FDA had asked applicants to supplement or amend their applications due to integrity issues, and also not the first time that the FDA had changed the TE code of the affected applications due to the integrity issues. However, this was a pivot from the past cases, in that here the driver of the action was extensive investigative analysis by the assessors. It was not solely based on the observations during the inspections. And that's understandable, because when we have intentionally compromised data, they will not always be apparent. They are very challenging to uncover. And site inspections may not provide evidence of intentionally compromised data. For example, when a CRO may be switching the study samples or analyzing the same set of samples and then swapping the data. This type of falsified data can sometimes become apparent when the assessors in the Office of Bioequivalence evaluate the totality of the information and all the evidence before them. Another pivot in this case was that all study data ever submitted from these two CRO facilities were rejected. This was in contrast to only the study data from specific time period in earlier cases involving integrity issues. Now, having covered the background, I want to talk about how the applicants responded to the information request letters and FDA's assessment of their responses. And you will see few common themes becoming apparent in the responses from the applicants. So first, many applicants, they responded with their plan to submit repeat studies. Some applicants, they asked if they could reanalyze the study samples at another CRO because subject samples were still available and stored at the impacted CROs. Unfortunately, this proposal was not acceptable to the FDA. That's because the integrity of the stored samples could not be assured since the CROs had engaged in creating false data in their studies to start with. Then there were another set of applicants. They disagreed with the FDA's position that the studies were inadequate, and they did not think that the studies needed to be repeated. And because they indicated that the sites or the studies were inspected earlier by the FDA, and no concerns were identified and no issues were raised either at the clinical or the analytical sites. And in some cases, these sites were inspected on more than one occasion. And that's correct, that's a fact. However, FDA's assessment was that when data are intentionally manipulated, past inspections cannot provide us sufficient reassurance. And I already alluded to this point in my previous slide. So then during inspections, if there is absence of evidence, it does not necessarily mean evidence of absence when we are dealing with fabricated data. Now then, there were also some other applicants who conducted their own investigations or they engaged third-party auditors. And these investigations and the third-party audits did not find any issues with their specific studies. And they indicated that they had verified the clinical documents and also the analytical and the statistical data. However, here again, FDA had no confidence in the conclusions of these investigations and the audits. And I say this because for the same applications, our analysis had identified data anomalies. The issue here was that rather than doing a systematic assessment, only random checking of the data anomalies were performed during the audits. At least that's what was reported to us. So then none of the subjects identified by us as being falsified or pairs of subjects with overlapping profiles were included in their investigations or their audit reports. And then the next two themes, they are related to the BX rating. So some applicants, they agreed to repeat the studies, but they requested we change the therapeutic code back from BX to AB. And others, they made the same request, but they did not want to supplement their ANDAs with repeat studies. And then there were another set of applicants who cited concerns about the BX rating causing a potential drug shortage for their drug products because they held the major market shares. Now, in all these cases, FDA explained that removing the BX code could only be appropriate if we had made an error in identifying the application as relying on the data from these impacted CROs. Otherwise, the agency needed to treat all approved applications similarly, irrespective of their market share. So now let's look at this slide. It shows a tally for the status of the impacted ANDAs as of February 2023. And you may be interested in knowing how things turned out. So here we can put the impacted applications into 4 buckets. The first bucket includes percentage of the applications, both approved and pending, for which the studies were repeated and submitted for review. The next one shows the applications that were withdrawn. The third bucket includes applications for which the applicants told us, they informed us that the studies had failed to meet the bioequivalence when they were repeated. And then the last one is a miscellaneous bucket. It includes applications for which extensions were granted with adequate justifications and documentation. For example, some applicants, they may have cited challenges with procuring the reference product, or some other applicants, they had to go through the process of requesting the agency to assign a new reference standard because it was not available, no reference standards were available on the market. And this would have caused some delays for them to start the studies. In addition, we also have to remember that this was all happening during the COVID period. Now I want to pivot to the impact of these actions on the key stakeholders. First, the patients, and then the applicants. As far as our knowledge goes, there was no additional burden for the patients. No drug shortages were identified. And all throughout, including the time period of the announcement of the actions, the agency had remained vigilant and monitored the potential for drug shortages for these impacted drugs. And we had also continued to monitor and perform post-marketing safety analysis and evaluate any safety signals or lack of effect signals for these approved drugs. And we did not note any such signals. Now, you can imagine all this requiring massive collaborative efforts and cross-office coordination, and it caused a tremendous increase in our workload, including the work to be done to assess large volumes of new data of the supplements and the amendments. For the applicants, when we did the market share impact analysis, we did indeed find the decrease in the market share for many generic drugs. And also, a substantial number of ANDAs were withdrawn by the applicants, either for business reasons or a non-bioequivalent product. Since we don't have all the information, we cannot tease out what was the reason for the withdrawing. Thinking about the key lessons from this experience, one lesson that is very clear and it stands out is that there is no place for falsified data in FDA-regulated products. FDA will take action against, within its authority once it becomes aware of the falsified data or information in the submissions. And also, depending on the nature of the violations, it may impact all work ever done at the testing facility site, as was in this case. The other lesson we can think about is that the cost at the back end can be costlier than the cost at the front end, because when we have to repeat large number of studies, it can be a huge financial burden. So sponsors need to provide a greater oversight for the outsourced portions of the applications. Also going forward, the agency and applicants need to stay vigilant, at least to the known patterns of data manipulation. And I have provided here a few of them. For example, preferential chromatographic reintegration. I will say we used to see a lot of manual reintegrations in the past, specifically for failing QCs to bring them into passing. And also samples, which were the CMAX samples, would be preferentially be reintegrated so that the ME study would be met. However, we remain vigilant on this aspect, and more recently, we do not see as many manually reintegrated chromatograms. Some other examples would include preferential sample reanalysis, falsification of lab date records, sample substitutions, or slipping in fabricated values for a couple of samples in the statistical analysis, especially the CMAX samples. As for the next steps to mitigate the risk, I will first share with you at least two measures that the Office of Generic Drugs is undertaking to identify unreliable data. So the first one is enhancing data analytics and visualization tools to identify data integrity issues. Here, we are leveraging the functionality of the software tools to automate detection of discrepant and atypical data in the submissions. And we have already adopted some of these tools as an integral part of our study assessment now. The other is working collaboratively and in partnership with our internal and external stakeholders. Internally, this includes cross-office multidisciplinary team approach between our assessors, the OSIS investigators, and our statistical experts. External collaboration includes global health authorities, the counterparts of the global health authorities. And we have established a process, and we hope that sharing the information and the data about the concerns during the assessment among the regulators at an early stage would bring more awareness and also more data points for the agency's assessment, and this could potentially shorten the time of taking the action. What are some of the steps that sponsors and CROs could take to mitigate the risk of compromised data? For the sponsors, there are a number of guidance documents from regulatory agencies and also non-regulatory bodies, all providing very similar recommendations and laying out the expectations of the sponsor's responsibilities. By being diligent about site selection, monitoring the study conduct, and confirming the reliability of the study data for the outsourced portions, sponsors can meet their responsibilities for assuring the integrity of the data in the submissions. For the CROs, to ensure the integrity of the data that they're generating at their facilities, they would need to establish a robust quality management system. And more importantly, the leadership would need to support and promote a culture of quality within the organization. And then lastly, sponsors and site personnel should consider reporting fraud and data manipulation in the study conduct. Sponsors may come across this type of information either during their monitoring or study audits. And you can send this information through the mailboxes noted here or include the information in the submission. More recently, some sponsors have reported to us unreliable data that were identified during their audits. And we greatly appreciate that type of transparency. On this note, I will conclude my presentation. Thank you for listening. Before I go, I have 2 challenge questions. So here's question 1. The market share of generic drugs were not impacted by FDA's actions. True or false? The answer is false. As I mentioned earlier, when we did the impact analysis, the market shares of many of the ANDAs, many of the drug products, the generic products, were impacted. Question 2. Sponsors are expected to verify the accuracy of reports submitted by a CRO in support of marketing applications. True or false? The answer is true. The sponsors are responsible for assuring the accuracy of the data and the information that they include in the submissions to the agency. With that, I thank you again, and I will be joining the panel where I'll be happy to answer any questions for you. Thank you.
Hello, my name is Pallavi Nityanandan and I serve as the Director for the Compendial Operations and Standard Staff, and today I will be presenting on the risk factors for benzene contamination in drug products and will be specifically talking about the excipient carbomer. This talk will begin with providing background on the topic, clarifying interpretation of ICH-Q3C guidelines on residual solvents, discussing USP carbomer monographs, as well as FDA recommendations to USP on the revision of carbomer monographs and recent USP revision proposals for carbomer monographs. As many of you know already, benzene is a known human carcinogen that causes leukemia and other blood disorders. Recently, FDA has posted information on our website to inform stakeholders of recent recalls due to benzene contamination. The link to the information on the website is provided at the bottom of the slide. The benzene contamination may be related to inactive ingredients such as carbomers, which are used as thickening agents in drug products, isobutane, which is a spray propellant, or any other drug component made from hydrocarbons. The information on the website also reminds drug manufacturers of the cGMP requirements based on which they must establish scientifically sound and appropriate specifications and test procedures to assure drug components, both active and inactive ingredients, and finished drug products conform to appropriate quality specifications as outlined in 21 CFR 211.84 and 211.160. The manufacturers are also required to test their raw materials and finish drug product batches according to 21 CFR 165 prior to release to ensure that they meet appropriate specifications for identity, strength, quality, and purity. FDA also recommends using risk assessments as a tool to determine if manufacturers have appropriate specifications, test methods, and controls to ensure drugs are free from contamination, and also evaluate the possible presence of benzene in their drug products and components, including active ingredients and inactive ingredients. So USP-NF currently has 8 monographs for various grades of carbomers. The numbered carbomer monographs indicated in the portion of the table with the orange shading, which is namely the 934, 934P, 940, 941, and the 1342 are the ones which are manufactured using benzene, and the individual monographs have benzene limits that significantly exceed 2 ppm as compared to the other 3 grades of the carbomers. Now we will discuss the interpretation of ICHQ3C residual solvents guidelines, regarding which there appears to be some confusion. As per ICHQ3C guidelines and USP General Chapter 467, Class 1 solvents should not be employed in the manufacture of drug substances, excipients, and drug products because of their unacceptable toxicity and their deleterious environmental effect. Exception for drugs considered as a significant therapeutic in advance, and this is a very important concept, so please make note. Only if a class 1 solvent's use is unavoidable in order to produce a drug product with a significant therapeutic advance, then their levels should be restricted as shown in the Table 1 of the ICHQ3C guideline, unless otherwise justified, which happens to be 2 ppm, which is listed for benzene in the guideline. Just restating that again, only if a drug product is deemed to be significant therapeutic advance and the use of a class 1 solvent is unavoidable in its manufacture, then the level should be restricted as listed in the guideline. Please note that sunscreens and hand sanitizers, which are the subject of the recent recalls, these are not considered as significant therapeutic advance, nor is the use of benzene as a solvent unavoidable in their manufacture. Continuing the discussion on ICHQ3C, even if it meets the criteria we have just discussed, when the use of the class 1 solvent is justified, then these carbomer monographs, which are the numbered carbomer monographs 934, 934P, 940, 941, and the 1342, allow residual benzene levels that are significantly higher than the 2 ppm permitted by Table 1 of the ICHQ3C and the USP 467 General Chapter on Residual Solvents. So very important point, please make note that the ICHQ3C guideline and USP 467 are not to be interpreted as recommending that controlling benzene at not more than 2 ppm at the drug product alone is sufficient in the absence of significant therapeutic advance. So that seems to be a point of major confusion, so hope you will take that away from this presentation. So FDA has had conversations with USP and made the initial recommendation to omit these monographs as early as February 2020, and we really recommended the deletion or omission of the numbered carbomer monographs beginning in 2020, and we have had several conversations on that. But an official letter was sent in November 2022, summarizing the previous discussions, reiterating the known health and safety risks associated with benzene, and also we recommended that USP make the monograph omissions effective no later than 2025, which would also provide sufficient time for the implementation of this change for stakeholders. So following our communication and recommendations to USP, they have posted a notice of intent to revise on November 18th, 2022 on their website, informing stakeholders that they intend to omit the numbered carbomer monographs with a targeted official date of August 1st, 2025. So FDA recognizes that industry stakeholders may have questions on the reformulation and how to report the changes to the agency. So we plan to publish a guidance for industry on products with benzene containing carbomers, recommendations for reformulation, which will advise on reporting categories for various dosage forms based on risk considerations for switching to another carbomer with similar physicochemical properties. So Hopefully we'll get to see that soon. And FDA encourages early implementation of reformulation if using carbomers 934, 934P, 940, 941 or 1342 prior to the proposed monograph omission date of August 1st, 2025, which means you don't need to wait until the monographs are going to be omitted. You know that the change is coming, so you can implement the change reformulation as soon as your company is able to. That brings us to the end of this presentation. Thank you so much for your attention.
Hello. Thank you for that kind introduction. My name is David Carey. I am the Office Director of the Office of Testing and Research, which is in the Office of Pharmaceutical Quality. And I'm going to be speaking about some studies we did in the lab around mitigation of nitrosamine formation in pharmaceutical formulations. Before I begin, I just want to say that patients expect safe and effective medicines with every dose they take. And pharmaceutical quality is assuring every dose is safe and effective, free of contamination and defects. It's what gives patients confidence in their next dose of medicine and what we strive to provide. So in 1978, a group of World Health Organization experts suggested something called a NAP, or nitration assay procedure, as a general in vitro test under some standard conditions, which is 4-to-1 nitrate to drug at 37 degrees and pH 3 to 4 under aqueous conditions. And to study the nitration ability of drug substances, and this was really kind of targeted toward conversion within humans. And the conditions they settled on are really quite optimized for the formation of nitrosamine. It's quite favorable for the formation of nitrosamine. And the two drugs in our study, drug A and B, the first one of these formed a nitroso drug substance-related impurity pretty fastly under NAP conditions. And the second one, which had a process-related impurity, which was dimethylamine, under NAP conditions formed NDMA. An important note about using these NAP conditions is that they're favorable, as I stated, for nitrosamine formation. But drugs containing reactive amines may never experience the conditions, these optimal conditions. So it doesn't necessarily mean if you get a positive result with or nitrosamine formation under NAP conditions with a given drug that will actually happen to the drug. So so to form nitrosamines, you need a source of a nitrization. You need a source of nitrite. And nitrite is one of those sources. And then you need a vulnerable secondary amine. And there's a recent publication about the LASA database, which is in the Journal of Pharmaceutical Sciences, published in 2022, where they measured the level of nitrite in parts per million in a number of different excipients that are commonly used to formulate drug products. There's some of them are listed here on this chart. Crosspovidone, magnesium stearate, povidone, microcrystalline cellulose, lactose, hypermellose, crosscarmellose, and cornstarch. And you can see each of them has varying amounts across all the lots and batches that they tested, with crosspovidone having the highest amounts, close to 10 part per million. And of course, when you would make a given formulation, given solid oral dosage form, you would mix these together in some proportion. And in a typical formulation, an average amount of nitrite present is about 1 part per million. So in most drug product formulations, you're going to have some amount of nitrite present. And to form small amounts, trace amounts of nitrosamines in a drug product, you really, of course, don't need much of the nitrite around. So this is the excipients are a source of potential nitrosation progenitors. And so in this study, we have the two drugs, and we know that in marketed drug products, these two drugs form nitrosamines, and that formation was dependent on, was in some formulations and not others. And so in the lab, we mimicked drug product manufacturing steps, and we also spiked in excess nitrite. We exposed them to heat and humidity, and we manufactured tablets in-house that either contain or did not contain antioxidants, or contain or controlled or had their pH of the formulation adjusted or not. And our goals from this study are to widely share the findings, and I'm kind of doing that today here, and hopefully to provide some examples of strategies that could be used to mitigate the presence of these in drug products. So this is the OTR manufacturing process. We have one of the two drugs. It goes through a mixing step, and granulation, and this is the step where we could spike in nitrite or dimethylamine if we wanted to. It goes through semi-drying and drying using a fluidized bed drying method. We have a granule formation step, and then ultimately the tableting. So here's some of the results with the first drug, which formed an NDSRI. And these drugs that we were put on stability for 6 months. And we made measurements across these 6 months. This particular set of experiments was done at 25 degrees C and 60% relative humidity, and then the amounts of the NDSRI that formed over that time was monitored. And there are 3 different antioxidants: ascorbic acid, caffeic acid, and ferulic acid. And then also we applied either acidifying or using sodium bicarb to make the formulation basic in this panel here. In each case, the blue line is the control, where we have added no amount of antioxidant or no change. It's the standard formulation. And you can see that if you do nothing under these conditions, that you get an increase in the amount of NDSRI over a 6-month time period up to about 200 part per million. But if you have ascorbic acid either at 1%, 0.5%, or 0.1%, you get a pretty big inhibition of the formation of that NDSRI. Caffeic and ferulic also have impact, but not as much as ascorbic acid in this case. And actually, the most dramatic difference was using the sodium bicarb, which basically didn't show any formation. In the other drug, and here we were getting NDMA formation. This is this is color coded through the 3 antioxidants: ascorbic acid, caffeic acid, and ferulic acid. And here, the blue is the initial measurement. The orange is day 30. The light blue is day 60. The darker blue is day 90, and the purple is day 180. And you can see in this case, actually, the ferulic acid had the this one formulation that showed the most formation of NDMA was spiked with 100 part per million of nitrite. So a really big excess of nitrite was added to this formulation. But what you can see that's from these data, if you look carefully, is that when we spike in a lot of nitrite, the ferulic acid in this case seemed to have the most inhibitory activity compared to caffeic and ascorbic acid, which had kind of a similar level of inhibition. The other thing you can see is that even when it is formed like at day 0, when it was formed in the manufacturing step, that it decreases over time, towards 6 months. So a lot of interesting data in here. Of course, I don't have time to break it all down. Hopefully, we'll be sharing this in more detail soon. But I did want to kind of show these preliminary data, and then some of the preliminary observations that we can make from them. In terms of antioxidants for the first drug substance, which the highest inhibition of NDSRI formation among the antioxidants was observed as ascorbic acid, which was greater than caffeic acid, which is greater than ferulic acid. For the second drug, however, the rank order potency was reversed. Ferulic acid was greater than caffeic, which was about the same as ascorbic. And so the antioxidants need to be fit for purpose, and their effectiveness depends on the drug substance and the manufacturing and the formulation. An increase in antioxidant concentration improved the NDSRI mitigation. So for pH manipulation, a set of conditions, to no one's surprise, facilitated nitrization reactions. Maintaining a neutral pH of the drug product served as a protective strategy against nitrosamine formation. And so an alkaline modifier, sodium bicarb, had the most effective inhibition of NDSRI formation and NDMA formation for the two drug substances tested. And also heat and moisture control would be an important factor. The data suggests that formation of nitrosamines was greater under conditions of elevated heat and moisture, and that the main nitrosamines, or the main progenitors for making them, were introduced or formed during the drying step of wet granulation. This might be due to the presence of secondary amine and oxides of nitrogen in the wet mass, which was then converted to a nitrosamine impurity during drying. So in general, antioxidants and pH mitigate the formation of nitrosamines in these two drug formulations that we studied, and the effectiveness of that mitigation strategy was product dependent. I want to acknowledge a huge number of people that are involved in this, particularly Jan Yang and Dia Shaklia. Most of this work was done in the White Oak labs of OTR, and then the St. Louis lab helped in terms of the testing of all these samples. And of course, my colleagues in various discussions around all things nitrosamines. And thank you for your attention.
Thank you all of our presenters for another group of great presentations. We'll move into our QA panel session at this time. If you haven't had a chance to enter your questions into the Q&A chat pod, please do so now. We'll answer as many questions as time allows. We've already got some questions coming in, and the first group of questions, the first two questions, will be addressed to Jonathan Resnick. And here is the first question. When will new Type 3 and Type 5 DMFs be considered for pilot in version 4.0?
Great question. So once FDA starts accepting version 4.0 submissions, you will be able to send in a new Type 3 or Type 5. Really, you can send in any application type as long as it's a new application. So it could be an ANDA, if it's a new one, or it could be a DMF. It could be a Type 2, 3, 4, or 5. The key is you'll need to wait until FDA announces that we are accepting those voluntary 4.0 submissions, which in all likelihood probably won't be until early next year. We will update the FDA data standards catalog with the eCTD 4.0 standard, and in that catalog it will say the date that we start supporting 4.0. So you'll be able to look there as well as our eCTD website. And I noticed you have the word pilot in your question. So you might be having in mind maybe sending in a test DMF submission to make sure everything's working okay. We do plan to open the door to submit test submissions using 4.0 later this year, and we'll have information about that on our eCTD site.
Thanks for responding to that question. We do have another similar question about testing out eCTD version 4.0 capability before sending in a real submission. Are we able to submit a sample to FDA for feedback?
Absolutely. Our plan is definitely to allow industry to submit sample 4.0 submissions so that everyone can make sure that these submissions are being published properly into the standard before an actual real live submission comes in. So we have a current sample submission process set up today for eCTD 3.2.2, our current version. You can learn about that process on our eCTD webpage. For those of you that don't know how to get to our eCTD webpage, it's quite easy. It's fda.gov/eCTD. Now, what we're going to do, and I just kind of alluded to that in the last question, is later this year, we're going to update our webpage that pertains to how to submit a sample eCTD submission. So it includes specific information about how to do the 4.0 sample. I don't have a specific date yet, but look for it to happen around the September timeframe of this year.
Thank you for responding to that question. Moving on to our next panelist, we've got a few questions that came in for Seyoum Senay, and here is the first question. Does the CDER NextGen portal replace the need to use the old ESG gateway and the need to have security keys?
Great question. So does the CDER NextGen portal replace the need to use ESG? The answer will be no. The CDER NextGen portal does not replace the electronic submission gateway. The portal is used to submit non-eCTD submissions, which are not required to have an eCTD format. Except in some cases, if, for example, for research IND, yes, we can accept the submission via portal. Hope that helps. Thank you.
Thank you for responding to that question. We have another CDER NextGen portal question that came in for Seyoum Senay, and here's the question. Can CDER NextGen portal be used for requesting IND application number as well?
Another great question. I'm going to repeat the question one more time. Can the CDER NextGen portal be used for requesting IND application number as well? The answer is yes. An IND sponsor or the U.S. agent can register and submit a request through the CDER NextGen portal to obtain an IND application number. If you have any question, please let us know. But the answer is, in short, it's possible to request IND application number via portal. Thank you.
Thank you for responding to that question. Moving on to our next panelist, we have a couple questions that came in for Dr. Nilufer Tampal. And here is the question. If the CRO is identified swapping the data to fabricate the BE results, will the CRO be barred from other ongoing studies with NDAs and ANDA applications?
Yeah, hi. Thank you for the question. I think to the best of my knowledge, we don't have regulations that speak to the debarment of a CRO. Maybe you may be thinking about our regulations for debarring a clinical investigator, but the same similar regulations for the CROs we don't have. Thank you.
Thank you for responding to that question. We have another question for Dr. Nilufer Tampal, and here's the question. Does FDA perform the reanalysis to conclude the CRO's fabrication data, or is it based on statistical analysis of raw data, or just a review of the data?
Yeah, here I think what we do conduct the statistical analysis of the submitted data, and that's a routine part of our assessment. And in that process, say we observe some concerns. For example, sometimes the statistical output of our analysis may not match the applicant's analysis. Or the assessors may notice some discrepancies in the values in the data sets in the submissions. Then additional analysis or the investigation would be undertaken. And the steps which an assessor may consider will all be driven by the nature of the concern or the issue before them. Thank you.
Thank you for responding to that question. Moving on to the next panelist. Our next group of questions is directed to Pallavi Nithyanandan, and here is the first question. Is it sufficient to restrict the level of benzene at 2 parts per million in the drug product and continue using ingredients and excipients that contain benzene?
Hello, thank you for that question. So the answer would be no, and as explained in the slide 6 of the presentation, only under narrow circumstances, if a product meets the 2 criteria, one of which is that the use of a class 1 solvent is unavoidable in order to produce the drug product with a significant therapeutic advance, then that's the only when those 2 conditions are met, then you may be allowed to use ingredient at containing benzene. Otherwise, I think they should be avoided. That's what the ICHQ3C guidelines specify. Thank you.
Thank you for responding to that question. We do have another question for Dr. Nithyanandan, and here's the question. Does industry need to wait until USP's proposed monograph omission date of 2025 to switch to carbomer that's now manufactured using benzene?
So the answer is you don't have to wait because this is a change that is for the improvement of public health, so you don't need to wait, and early adoption is very much encouraged as soon as you're possible to switch from, it is possible for your firm to switch from the benzene containing carbomer grade to the non-benzene containing carbomer grade. You should go ahead and implement the change. Thank you.
Thank you for responding to that question. Moving on to our next panelist. We just got a few questions in for David Curie, and here is the first question. What is the chemical mechanism for the antioxidants to inhibit the nitrosamine formation?
Well, it's a very good question. I think if you were talking about these reactions in the in solution, you know that these things like ascorbic acid are known to be free radical scavengers. So that's potentially a route, and also in terms of neutralizing nitrite that's present, that's a known reaction where you pretty fastly form dehydroascorbic acid, and so, but of course, you realize that what's going on is in the solid state in these tablets, and so it's what the real mechanism is still a question, and how the antioxidants are doing the inhibition is also kind of an open question, but it's likely either by impacting the nitrite that's present, giving it another route to which to react, or by some sort of free radical scavenging mechanism. Thank you for the question.
Thank you for responding to that question. We've got another question just came in from Dr. Curie, and here's the question. We see that you use the likes of caffeic acid and ferulic acid in the studies. These additives are not in the IID. Is there a possibility that sponsors can start thinking of using these based on the safety data in the public domain?
Thank you for that question. So we chose the antioxidants for the study because they were kind of in series, ascorbic, ferulic, caffeic, and ferulic, and they had been used in other published work, but we did realize that two of them are not IID, and you'd probably be better off sticking with those, at least in initial work, until there's some sort of agreement otherwise. Thank you for the question.
Thank you for responding to that question. We'd also like to welcome to our Q&A panel Dr. Li-Kent Liang, who's the branch chief in the Division of Liquid-Based Products 2, the Office of Life Cycle Drug Products, and we just had a few questions come in for Dr. Liang, and here is the first question. If the residual benzene level in a carbomer excipient conforms to the 2 ppm limit specified in the USP monographs for carbomer polymers, can I use it for any products without any additional considerations in terms of residual benzene?
Thank you for the question. My colleague Pallavi has already provided answer to a similar question earlier. I'd like to further comment that if the use of such a carbomer is justified from drug product quality perspective, USP tests and limits are considered minimum requirements. Additional consideration may need to be taken depending on the drug product involved. For example, if the drug product has a very high dose, you will have to take the dose level into consideration in assessing the impact of the benzene level in excipients and in other components of the product. And you may need to implement proper controls that may be stricter than what the USP monographs describes and provide justification for your control strategy. Thank you.
Thank you for responding to that question. We do have another question for Dr. Liang, and here is the question. Where can we get more guidance regarding the expectations when changing a carbomer manufactured using benzene as a solvent to a carbomer manufactured without the use of benzene?
Thank you for that question. As Pallavi mentioned earlier, there will be a guidance published to provide recommendations for various scenarios for carbomer change. In addition, a firm can engage the agency for discussion and advice. For example, a generic firm can submit a control correspondence, contact the project manager, or submit a formal meeting request for a complex drug product, depending on the stage of your application or the product development. Thank you.
Thank you for responding to that question. Moving back up to the top of our panel, we've got a couple more questions that came in for Jonathan Resnick. And here's the next question for Jonathan. I am new to eCTD and would like to know if FDA publishes a listing of all headings available in the eCTD.
So the answer to that is yes. On our fda.gov/eCTD webpage, we have a eCTD submission standards catalog. And one of the documents there is called the Comprehensive Table of Contents, Headings, and Hierarchy. And that provides a listing of all of the predefined standardized headings in modules 1 through 5. So all those harmonized headings, especially between modules 2 and 5 through ICH, are all listed there in that specification document for you to be able to access. So hopefully that helps answer that question.
Thank you for responding to that question. One more question just came in for Jonathan Resnick, and here is the question. You mentioned that FDA plans to accept voluntary eCTD version 4.0 submissions within the next year, but when will version 4.0 be required?
So short answer to that question is it's too early to say. So we first need to open the doors and start receiving those voluntary 4.0 submissions for new applications and make sure that the systems are running smoothly and that there are no major issues. After we find that things are working well, we will then open the door for people to transition older applications. So if you have maybe a drug master file that you originally opened up using eCTD 3.2.2, and now you want to start making submissions in 4.0 to it, that's something we're looking to allow industry to do probably about a year after we initially open the doors to providing a new application in 4.0. And once we're at that point, then FDA will determine the date that the 4.0 standard will be required, and then we will publish an FR notice to alert everyone of when that will be, and we'll put that date in the FDA data standards catalog and also make it prominently displayed on our FDA eCTD webpage.
Thank you for responding to that question. We've got just over a minute and a half left. I have another question that came in for Dr. Nilufer Tampal, and here's the question. Will FDA honor a sponsor's monitoring report as a justification that there were no data integrity issues for our study?
Yeah, that's a challenging question, and I thank you for even bringing it up. So, I'm going to assume here that this question is related to a study that is outsourced to a CRO, and the sponsor is providing the oversight through the monitoring. So now whether a monitoring report will be accepted as a justification, that would boil down to two things. First is the nature of the integrity issue. What I mean is the manner in which the data may be manipulated, whether that's very systematically spread it out, how egregious it was. So that's the first thing that needs to go into the consideration. And then the second is our level of confidence in the monitoring report. So we may need to be able to see and assess the thoroughness of the monitoring, how encompassing it was, whether it covered all the relevant aspects. So, but then in your judgment, if the monitoring report can serve as a supporting document, then you should submit it. And it may add to our overall assessment as we go through all the available information and evidence. It can be considered as a supporting piece of information, but because of the reasons I outlined, it is unlikely that it will serve as a standalone or a sole piece of evidence. Thank you.
Thank you for responding to that question. We are just about out of time, but we did have one question that just came in for Dr. David Curie about nitrosamines. And here's the question. what formulation strategies can be used to mitigate nitrosamine formation in drugs?
Well, I think the pH adjustments were the most impactful for our studies with these two drugs, and of course the addition of antioxidants. And you know what? One of the things I want to point out is we chose an acidic form of ascorbic acid and these other antioxidants to use. You know, if you use the sodium salt of it, you might do better, right? You know, so we learned something in these studies about, you know, using the sodium salts might be a better approach because it combines both antioxidant capacity and pH control. So that's kind of seems to be the two most important things. Thank you for the question.
Well, that's all the time we have for our questions and answers in this session. We certainly a huge thank you to all of our panelists and presenters over the past 2 days for the great presentation and answering a huge number of questions that came in. Just a few bit of housekeeping items. That recordings will be posted in approximately 5 to 7 business days. So look for those recordings on fda.gov/CDER-BIA. Remember again that certificate of attendance to support continuing education credits for SOCA RAPS and SQA and the CE claim codes for continuing education for healthcare providers, physicians, pharmacists, and nurses are available for 2 weeks. And remember, that deadline is April the 27th. Now it's our pleasure to welcome back Dr. Lei Zhang to share some closing thoughts. Welcome, Dr. Zhang.
Thank you, Ray. Good afternoon, good evening, and good morning to all the participants across the world. It is at the end of this year's Generic Drug Forum. I hope that you, like me, enjoyed every session of the past 2 days of the forum. In closing, I would like to thank all the participants for joining us from across the globe and making this 10th anniversary of the Generic Drug Forum a success. The past 2 days have been filled with valuable insights and practical advice on the ANDA assessment process, as well as updates on timely topics such as GDUFA III enhancements, product-specific guidances, industry meetings throughout the product development and life cycle, best practices and tips for ANDA submissions, policy, and information technology, just to name a few. It is encouraging to see the continued interest from our stakeholders in this forum, and we are committed to providing clarity and fostering collaboration with the generic drug industry. We had more than 3,600 people registered from more than 100 countries, and nearly 2,000 people participated via Adobe in the past 2 days. It took a village to organize and run this forum. I appreciate the efforts of the speakers, panelists, and the subject matter experts who worked tirelessly to deliver informative and engaging sessions. I also would like to extend my gratitude to our SBIA colleagues, Brenda, Ray, and others who moderated the sessions and coordinated behind-the-scene efforts. Finally, I would like to invite you all to join us for the upcoming SBIA webinars on the M13A draft guideline on May 2nd and on GDUFA III scientific meetings on May 15th to learn more about these topics. In addition, FDA also going to host 2 hybrid workshops with the Center for Research and Complex Generics, CRCG, on next week, April 20th to 21st, and also on May 10th, respectively, on alternatives to comparative clinical endpoints and pharmacodynamic bioequivalence studies for generic orally inhaled drug products and drug device combination training. I believe those topics will be of interest to this audience. We are also hosting our annual GDUFA Science Research Workshop, public workshop, on May 11 to 12 at the White Oak campus. it will be in a hybrid mode, have both in-person as well as online option for people to attend. We hope you can join us for these informative events. Follow us on the generic science research website or FDA meetings, conferences, and workshops website for future events that may be of interest to you. With that, I thank you all for your time and participation, and I look forward to seeing you again in the future. Thank you.